Hello and welcome back to Quick and Dirty Med School. Today we will be going through the third week of development. So the third week starts up with gastrulation, and the process of gastrulation is creating the trilaminar embryonic disc. The three layers are the ectoderm, the endoderm, and the mesoderm. All the embryonic tissues and axial orientations are established during this time. The ectoderm, just as a brief overview, is the central nervous system and the epidermis, along with various other structures. But the mesoderm primarily makes up the connective tissue, the viscera, the cardiovascular system, and vessels, along with the reproductive organs. The endoderm is the component of the alimentary canal, which means your mouth, your esophagus, down to your stomachs and then intestines. And then the germ cells are its own separate category because they are the components of the sperm and the egg. As you can see here, gastrulation begins with the blastula, and then the blastula will, first form like this hollow blastocele, and then it invaginates these cells in, forming the structure called the archenteron. Pardon me, I can't really pronounce these very well, but okay. But more importantly, you see these like cuboidal, and then they become columnar, and they invaginate in, and then it folds over, creating the layers. And then this little pore here is the blastopore, and eventually it's closed over with the mesoderm, and then the ectoderm will close over all of that. So the primitive streak is like the precursor to our spinal cord, and in week 3 the primitive streak develops caudally on the dorsal aspect. So it develops first on the caudal end, the head end, on the dorsal or the back side of the developing embryo. It results from the proliferation and movement of the apoplastic cells, and the cranial end proliferates and forms the primitive node, which is continuous with the primitive groove. And then there is also a primitive pit within the primitive node. And these are all formed by the ingression of the epiblastic cell. So the groove forms because they go inwards. And the primitive streak and the epiblast cells are pluripotent. So that means that these cells can they can differentiate into whatever other cells of the body. So the cardiogenic end, this is where the heart will eventually develop. The precordial, or like the oropharyngeal membrane, is where the future mouth is. The nodal cord will eventually be continuous with the primitive groove, but right now here's the primitive pit inside of the node into the groove, and then here's the cloacal membrane, which will be the future anus. The cells will leave the deep surface of the primitive streak and make the mesenchyme. So the mesenchyme makes mesoblasts, which are the undifferentiated embryonic connective tissue. And then the epiblast will differentiate into the embryonic endoderm that will displace the hypoblast cells. The cells that ingress will become the endoderm or the mesoderm. And then the epiblast eventually will make the embryonic ectoderm. These were the ones that were displaced, right? And then the primitive streak eventually disappears by the end of the 4th week, and the umbilical vesicle that is formed by all of the layers eventually differentiating is also the yolk sac. So the nodal cord is when the mesenchymal cells migrate cranially from the primitive node, and it makes the notochordal process. The process will grow cranially between the ectoderm and the endoderm until it reaches the precordial plate and acquires a lumen, which is like an opening, making the notochordal canal. The precordial mesoderm is mesenchymal population of neural crest origin, and it is rostral to the notochord. Rostral is just another word for cranial, so it's going to be superior or cephalic to the notochord. And then the precaudal plate will eventually develop the oropharyngeal membrane, and by the third week, the intra-embryonic mesoderm separates the ectoderm and the endoderm, except for at the oropharyngeal membrane, the neuroenteric canal, and the cloacal membrane. So in those three sections, the mesoderm will not be attached. The mesoderm, yes, will not be attached, and the ectoderm and the endoderm can fuse. The neuroenteric canal makes the communication between the amniotic and the yolk sac cavities, and the floor of the notochord process merge with the endoderm. The cells will go through the apoptosis, and then the process becomes the notochordal plate. The plates will fold under, and then forms the neurotube. The nucleus pulposus is the remnant of the notochord, and the chordoma are malignancies of the notochord or the spinal cord. Usually they are malignant, and they can develop in every part of the vertebral canal. Here it is. It has even infiltrated the brain. See right here, should be, this should still be like gray matter, right? Okay, so let's go through the process. Here's the neuroplate, here's the notochord, the groove. Here, right here would be the crest cells. They would go in, cells from here will displace these cells, kind of making it rise and then invaginate. Here you go. Here's the neurocrest cells. They keep folding, and then and they will merge, and then right here, this tiny slip right between the neurotube and the epiblast will go through apoptosis, and then this will be the true neurotube. The notochord defines the primordial longitudinal axis of the embryo and gives it some rigidity. It provides the signals necessary for the development of the axial musculoskeletal structures and the central nervous system. And it will induce the overlying embryonic ectoderm to thicken and form the neuroplate. So here's 3D reconstruction. Here's a transverse section. You can hardly see it here, but look, here is the groove. And then here it is trying to invaginate. Here it is actually invaginated and starting to apoptose right there. So the Alantois develops by day 16, and it begins as a direticulum from the yolk sac, and it extends into the connecting stalk. The allantoic stalk will differentiate into the umbilical arteries, and the proximal part will persist in development as the urachus, and then eventually it will degenerate, but part of the Alantois will remain in the adult as the medial umbilical ligament. So the anterior-posterior axis is signaled by the cells at the anterior visceral endoderm, also known as AVE. AVE will send signals to express OTX2, L1M1, HESX1, Cerberus, and Lefty. All these are genes or transcription factors or growth factors, and these all code for head formation. The combination of the signal is important. These signals will tell what exactly to differentiate, because although these all kind of differentiate for the head, like Lefty, for example, will be coding for the left side of the brain, and and if it is mixed with another like more inhibitory signal, then the right side will develop. These factors all inhibit nodal at the cranial end. Makes sense. Nodal initiates and maintains the primitive streak, so that's more towards the dorsal end. BMP4 and the fibroblast growth factors will ventralize the mesoderm, while cordon, which is also activated by glucocorticoid, noggin, and golostatin will antagonize BMP4 to dorsalize the mesoderm into the nodal cord, the somites, and the somatomeres. So here are the somites, here's the nodal cord, here's the precordial plate, somatomere. Here's a few disorders. The glucocorticoid mutation is also, you can remember it by the acronym SAMS, which means short stature. Auditory canal atresia, which means they may not have a fully developed auditory canal. Mandibular hypoplasia, which means that the jaw is not fully developed. Look how regressed the jaw is and how much of an overbite the child has. The skeletal abnormalities, so the child can have like a shortened humerus, and the metaphysis of the long bones are flared, like right here in the femur, right here in the ulnar. And they also have fused joints. You can see the fused joints, the hands. HNF3 beta, it maintains the node and induces region specificity in the forebrain and the midbrain area. So this is an important code or gene for brain development, and in the absence, the embryos fail to gastrulate and lack forebrain and midbrain structure. These embryos are non-viable and they will not be able to survive past like week 4 of development. Caldo dysplasia, also known as Caldo regression syndrome, caldoagenesis, sacroagenesis. All of that basically means is there's a abnormal shortening of the body in the caudal region or in the sacral region. It just doesn't develop. You can see here's this child. The pelvic region, lumbar-ish region is abnormally short. The mutations, the syndromes will affect the movement of the lower body, such as like flexion of the lower body, rotation of the lower body. There's going to be anomalies of the lumbar and sacral vertebra. In some severe cases, no kidneys, no anus or urinary tract will develop, and of course this is fatal for the embryo. These children will not be able to survive. Agenesis of internal genital organs, except for the gonads, will also occur. Brachyury gene codes for the length of the embryonic axis. And so they believe that this gene is involved with this caudodysplasia. Another abnormality or defect is called syrenmelia, which is the fusion of the limbs. As you can see here, remember siren, mermaid, but here there's this one's less of a fusion and more like only one limb has developed, but here you can see the fusion because there are the two knees and then there's the feet. It also has vertebral defects, anal atresia, tracheo, esophageal fistula. These two are abnormally connected together. As you know, the trachea and the esophagus are separate structures, and they are also separated by a layer of like viscera. And then the renal and radial forearm will also have like abnormalities. This has an association with taking high levels of insulin during pregnancy. I don't believe that this is the direct cause, but this just has a very strong association. So teratomas are made out of the three layers: the ectoderm, endoderm, and mesoderm. And the sacrococcygeal teratoma are the remnants of the primitive streak. They're the most common tumor in newborns, and like 80% of them are manifested in females. And surgery is the only complete cure. So once the child has gotten surgery, it doesn't come back. It's not like malignant or anything, but as you can see here, it is very, it can be very, very large. So here are other, well, this is more like teratomas. This would be like just an external kind of teratoma. This one has some of it inside of the cavity itself as well. Type 3, it infiltrates even higher, as you can see. Type 4, it will it wouldn't protrude outwards. Instead, it would go inwards into like the anus or the pelvic cavity, in the maybe the uterus for the females. So the left-right axis is kind of tricky because PIT X2 is the master gene for the left-sidedness, and all we know is that it's inhibited by MAO on the right, and MAO is found more on the right side of the body. So what we're kind of assuming is that the right side of the body forms because of the absence of left-sided genes like PIT X2. The normal left-right positioning is called situs solitus right here. This is the normal. And then an alteration in HT signaling can result in situs inversus and dextrocardia, which means the heart is going to be like flip-flopped. Here's the normal heart. Of course, normally the apex of the heart will point to the left. Here it's going to the right. There's a couple different versions, I suppose, of situs inversus, because situs inversus is like the full flip-flop. Like all your organs are flipped. It's generally in a heterotaxy spectrum where the situs ambiguous or heterotaxy is when the organs are abnormally positioned, but that doesn't mean all of the organs. So it might mean like maybe only the heart was flipped, or maybe only the stomach and the heart was flipped, but it's not like every single organ. So the right atrial isomerism syndrome, or the right-sided heterotaxy, is involved with like no spleen. So there would be so in this particular syndrome, there would be no spleen development. There's just no spleen present. But on the left atrial isomerism syndrome, there's going to be like multiple spleens. As you can see in this MRI, here's the pancreas, here's the jejunum, and look, there's 3 spleens here. That's not right. We only need 1. Okay, so neuralation. The neuroplate makes the neurofolds, which close to make the neurotube, and this is all finished by week 4. The depressed mid-region forms the neurogroove, and the anterior pore and the caudal neuropores are where the neurotube communicates with the amniotic cavity. And the cephalic part will go on and make the brain vesicles, while the caudal part will develop in the spinal cord. The neurocrest cells will migrate once the tube has closed. So you can see here. The neurocrest cells are at the lateral border of the neuroectoderm, and the cell population undergoes epithelial to mesenchymal transition. So once the tube has been closed, it kind of like ingresses in and becomes mesenchymal. The cells will leave the neuroectoderm by active migration and displacement. like I said, to enter the underlying mesoderm. And the crest cells will migrate along two different pathways. Along the dorsal pathway, it will go to the dermis, and that will make melanocytes in either the skin or the hair. And the other pathway is the ventral pathway, where it goes through the anterior half of each somite to be a sensory ganglia or a sympathetic and enteric neuron. It can also go and make Schwann cells or cells of the adrenal medulla. Secondary neuralation is when the condensation of the central tail buds will turn into a solid mass, also known as the medullary cord. It will go through a process called cavitation to form a new lumen, and that lumen will merge with the neurocanal of the neurotube, making it like continuous. The lateral tail bud will undergo, as you can see here, segmentation to form the Caldo somites, and the caudal end of the notochord will grow into the sacrococcygeal and tail region. And this is all finished by week 8. So first, it is condensed by the the medullary cord, and then there's the cavitation, making a cavity, and then it degenerates, leaving only the somites behind. Just to go over again, the ectoderm, here it is. The outside forms the exoskeleton. It makes the central nervous system, the peripheral nervous system. It also is the precursor to the sensory epithelium of the ear, nose, and eye. Also creates the epidermis of the body, along with the subcutaneous glands, the mammary glands, and the pituitary glands. And oddly enough, it also forms the enamel of the teeth. Okay, defects. So, anencephaly is where the brain does not form. It's different from microcephaly, because microcephaly, that means that the brain is small and underdeveloped, but it's still there. This is when it's just not there at all. The only thing this poor thing has is basically just like a brainstem if it's lucky, but this is definitely not viable. Spinal bifida is when the secondary neuralation is not complete. And let's go back. Let's go back a little bit more. I don't like these slides. Oh, here we go. So the neurotube does not close completely, right? So it might end like right here, and then the child would be born with like bits of the spinal cord herniating outwards. Rachiaschisis is the complete non-closure, so it is like a step further than spinal bifida. So this is spinal bifida. Generally, there's only like one portion that is not closed, and then the spinal cord, along with like the meninges, will like bulge outwards. But here, it's not even closed at all. Look, all of this is exposed. Children can be born and be viable with the spinal bifida. It is, I don't want to say easily corrected with surgery, because it is quite a complicated procedure, but it is fairly straightforward. The child can go through the surgery and live a long, healthy life afterwards. But a child born with the rachiasis will not be able to live at all. The fetus is not even able to be viable past, I think, week 8. So, fortunately, this can all be prevented by taking folic acid. 400 milligrams of folic acid as a prenatal or during pregnancy, as your your pregnancy gestation supplements. And honestly, people aren't really sure how this works, like why the the spina bifida occurs with the absence of folic acid, but the prevailing theory is that the critical genes are unmethylated, so the tube doesn't close, and the folic acid will provide the methyl groups for the genes to be methylated. Okay, well, thank you for your attention and good luck studying.